In this module, we're going to be talking about macromolecules, and a good place to start would be just making sure we know exactly what a macromolecule is. We can get a pretty good definition of a macromolecule by taking the term and sort of breaking apart the meaning of the pieces. So if we look at this prefix macro, we see that in a lot of different places, and it essentially means either big or large. And then if we look at the ending of this term, molecule, a molecule is going to be formed when we have two or more atoms bound together. Okay, so essentially what we're talking about is a large molecule. This is going to be a structure that is comprised of many atoms bound together. And so really what we're doing is we're kind of building upon what we've talked about previously in terms of scale. We've spent some time talking about atoms, which kind of act as the individual building blocks of molecules, and how those atoms can be combined together or bound together to make molecules. Essentially, kind of what we're doing is we're just kind of upping the ante a little bit, and now we're talking about structures that are many atoms in size, those being the macromolecules. We're going to see that the macromolecules definitely differ in structure, but a lot of the different categories of macromolecules tend to have a repetitious structure to them, where the overall larger structure is comprised of putting together similar units that are smaller. We have terms for these smaller units as well as the larger overall structure. The smaller units that are put together or bound together are called monomers, and then the larger entity that they end up producing are called polymers. Now, the monomers themselves can be molecules of a variety of atoms, and they're not necessarily all identical, but they have a similar base structure. Kind of a good analogy here would be to think about like a beaded necklace or a beaded bracelet. When you make beaded jewelry, each bead is kind of an individual subunit that gets connected to other beads to make the overall piece of jewelry. So in this analogy, the monomers are kind of like the beads. And we know that when we're making jewelry, the beads don't have to all be the same. Variety sometimes makes the jewelry a little more interesting. And then the overall end product that you get when you put those beads together would be sort of your necklace, which is analogous to the overall polymer. Now, while the structure is going to vary amongst these different macromolecules, what we're going to find is that there are some features that we see over and over again. These are specific arrangements of atoms that are going to be present within the larger structure, and these are called functional groups. So functional groups are going to be these arrangement of atoms that we find, and they're going to have similar properties even though they might be attached to a different structure. So for example, and you don't need to memorize these functional groups, we definitely want you to start to kind of appreciate that knowing their properties might help you predict the behavior of a larger molecule. An example of one functional group is called a hydroxyl group. A hydroxyl group is an oxygen bound to a hydrogen, and then that's bound to some larger structure. So if we're looking at our schematic here, you'll see that there's this letter R. The letter R is a placeholder. This is where it would attach to that larger molecule. So simply, the R is a placeholder for something else. Hydroxyl groups tend to be fairly polar, so things that we know about polar molecules would tend to be true about that region of this molecule. Conversely, a methyl functional group is comprised of a carbon bound to 3 hydrogens, and then the fourth bond is to a larger structure. And that tends to be a very non-polar functional group. So as you can imagine, it would be very neutral. It wouldn't like to mix with water. And so we would expect that type of behavior from that section of the molecule. So again, the key here is that it can be beneficial to start identifying functional groups because even if you're looking at a larger structure whose behavior you're uncertain of, those functional groups are going to have similar properties even if they are attached to a larger structure, and you can start to predict how they'll behave. There are two general types of reactions that help us either comprise a macromolecule or help break it down. And both of them involve water, either adding water or taking it away. When we're trying to build a macromolecule, we need to link together monomers, and we can do this through something called a dehydration synthesis. Here's the idea. In this picture, the little individual green circles represent the individual monomers that we're trying to link. And what we find is that some of these monomers have hydroxide groups. That's what this OH circle is right here. And then others might have hydrogen ions attached to them that they're bound to. What we can do to link these together, because keep in mind, in order to link these monomers, we need these monomers to form a bond. Well, currently the monomers are bound to as many atoms as they're going to want to be bound to. So the only way that we're going to be able to have them bind together is to get rid of a bond they were previously making. So what we're going to do is we're going to have the hydroxide and the hydrogen ion, both those bonds will be broken so that they can come together to now form water. And then the bond that the monomers were making with that hydroxide and that hydrogen, now that that bond has been removed so that they can make water, they now make that bond with one another to make a larger macromolecule. So the way that I always remember this is I think of dehydration synthesis. Well, dehydration means that you don't have enough water, or there's a lack of water. So in this case, we are removing water, which you would expect if there was a lack of water, right? So we're removing water from these molecules. We're creating this water molecule by removing it from the structure. And then synthesis means to build. So we are building these molecules by removing a water molecule. The opposite true of the reaction called hydrolysis. So in hydrolysis, what we're going to do is we're going to add water. And if we think about this word, that can help us too. Hydro means water, and to lyse means to split. So we're using a water molecule to split a macromolecule into smaller subunits. Essentially, what's going to happen here is that one of the hydrogen ions is going to break off from the rest of the water molecule, creating a hydroxide group and a hydrogen group that are then going to split a bond in the previous macromolecule. So again, very much the opposite of what we just did with dehydration synthesis. So we can build macromolecules and we can break them down into smaller subunits by either adding water or taking water away from the molecule. This right here is going to be a pretty good roadmap for the upcoming videos. There's basically 4 major macromolecules present in living organisms that we're going to cover. For each of these macromolecules, we're going to talk about the structure and the function of that group. And they are carbohydrates. The monomers of carbohydrates are known as monosaccharides, and then the polymers come into different scales and are known as disaccharides and polysaccharides. We're going to talk about lipids. Lipids are kind of the interesting one here. If you notice, we don't have a listed monomer or polymer, and that's because this is the one macromolecule that doesn't really have a repetitious structure. So there are no monomers and polymers. Instead, they're characterized together by a similar characteristic, which is that they tend to be nonpolar. We're going to talk about proteins. The monomers of proteins are amino acids, and then those form together in chains to make proteins, which are known as the polymer. And then with nucleic acids, the monomers are nucleotides, and they're going to bond together to make polymers that are called nucleic acids. And again, the goal here will be for each one of these groupings to talk about the structure of that particular monomer, that macromolecule, as well as the function of that macromolecule within a living organism.